the main part of this presentation is really going through and outlining the current uses of genomics in beef seed stock herds, really focusing in on parent verification, genetic conditions, qualitative traits such as horn, poll, coat colour, and what's happening in the production traits, and particularly how does that feed into breed plan. So some of this information will be new to you. What I've done, because it's such a broad area, you could spend an hour on each of those topics. So what I've done is I think I've picked out the most interesting parts of each of those, and I'm going to highlight those. What I will say at this stage is you will have more questions needed after this, so, and some parts I might not fill the gaps for you completely in this presentation because of the time. Please make sure you call your associated SBTS and TBDS technical officer to follow up any queries you have, and we'll make sure we fill in those gaps that you might go away with tonight. What I'm also going to leave you with is do a bit of crystal balling at the end, saying where are things up to and what's the next situation, what's next for the genomic space for seed stock herds. That's probably the most important part of the presentation in a way, to know what's coming up from our knowledge of what's happening currently. So that's where we're starting off. So the first part of this presentation, as I said, is really talking about the theory and terminology with genomics and DNA. Genomics is basically the investigation of looking at the whole genome of an animal or the whole DNA sequence of an animal. So we really need to get in and start looking and talking about what is DNA itself, because a lot of this technology, all this technology we're talking about tonight, really is based around DNA testing or testing for DNA, in particular gene markers, etc. So first of all, DNA can be expanded out to deoxyribonucleic acid. That's just a scientific name, so you don't need to get too caught up about that, but it's called DNA. But it gives us a bit of an idea about what the basis and what DNA is made up of. To think about it from where it stretches from, we have an animal over here. This is an Angus bull, obviously, but you could have a Hereford or Limousin or a Brahman. It's the same thing, or a human or a dog. But here we're talking about cattle. We can then move down, and each animal is made up of individual cells. We know that, and anyone who's done basic biology at high school will know that at school. Within each of those cells, we then have a nucleus. And in cattle, the nucleus has the chromosome situated. The chromosomes, there's 30 pairs of chromosomes in a beef animal. I think in humans there's 26. Don't quote me if I'm wrong, but in cattle we have 30 pairs of chromosomes. And the chromosomes are made up of our DNA strand, so that classical double helix structure you would have seen quite regularly. So that's where it flows through from the animal to the cell, to the nucleus, to the chromosome, to the DNA. So to explain what a gene is, so we hear the term genes, gene markers and things thrown around, we really need to look at DNA in more detail. And you can see we've gone from the DNA there to really honing in and focusing on that DNA in more detail. Let's now break that down into a few different areas. DNA is made up of 2 strings of nucleotides, which really gives us that double helix or that twisted ladder structure, which most of you would have seen publicised around the place. Nucleotide comprises of a range of things, as it says there, phosphates, deoxyribonucleic sugars and nitrogenous bases. But really the nitrogenous bases are the main part that we utilise when we're looking at gene markers, and particularly for DNA testing for certain things, as we'll go through tonight. The nitrogenous bases can come in 4 forms: A, T, G and C, and always the A's and T's are combined together, and the G's and the C's. As you can see, if you look down there, the base pairs, you've got G's and C's and T's and A's, and that's the way they are always combined. And its estimate, and this is based on what I've read recently, approximately 3 billion nucleotides in a beef animal's DNA. So there's a lot of these nucleotides across an animal's genome, you would say. So the important thing about these base pairs, it's the combination or the sequence of these base pairs which really makes animals genetically different from one another. So in a beef animal, a lot of these are going to be the same, but there will be differences in these base pairs and the sequence of those, which gives us differences. And it's really where the technology really hones in on these differences in the base pairs and the nitrogenous bases to work out how animals might differ genetically and really start working out at that level. And a gene is a base pair sequence at a particular spot within the DNA that codes for a specific product. So for example, we might grab that part there and say, we know from all the reference populations and performance recording and DNA testing we've done, we know that part of the genome, that part of the DNA, and that combination of base pairs is associated with marbling, or associated with muscling, or associated with calving ease or something, or it's adding to that particular trait. So that's where it basically comes from, but we'll go into more detail about this as we go along. And other estimates we've seen is there's between 22,000 and 26,000 genes in a beef animal on that particular basis. So that's basically a explanation of what a gene is. It's a combination of those base pairs or a base pair sequence in a short segment. There's several forms of gene markers that we have to get our head around, and a lot of these you've probably heard in terminology reasons, but the first one is what we call a single nucleotide polymorphism, and that's a lot of words to get out of your mouth, but basically it's shortened as a SNP, SNP. And basically what it is it's two DNA fragments from different individuals contain a difference in a single base pair. So you can see there, these might be two different animals, the top animal's DNA and the bottom animal's DNA, and you can see that all the base pair sequence is the same apart from one spot, where the top animal's got a C and a G, and the bottom animal's got a T and an A. So it just differs in one spot for a SNP. And basically most of our DNA technology, particularly for production traits, but also moving into parent verification more and more, is based on this SNP technology, mainly because you can do a really good sampling across an animal's entire genome, whether it's at 50,000 points or 800,000 points or 100 points. You can use this SNP technology quite economically to do that. So you'll see a lot of the DNA technologies moving, if it hasn't already, towards this SNP-based technology. The other form of gene marker that we need to get ahead of is microsatellites, because this form of gene marker is still being utilised, particularly in the parent verification field. And it's basically where microsatellites, what they look for is parts of the genome where we have a repeat of the base pairs over time. So you can see here, in this part of the segment of the genome, or the DNA, we have C's and G's and T's and A's repeated one after the other, and that gives us a microsatellite as such. And this is really the basis for traditional parent verification for DNA technology, and it's still used a lot today in parent verification. We'll talk more about that in a second. But you can also use SNPs in parent verification now, which we'll talk about. The other one I want to talk about, and I've actually only really got my head around this today after talking to Emily Piper, or indirectly to Emily Piper from UQ Animal Genetics Lab, is another form of gene marker which we now call indels, which is basically the basis for genetic condition and defect testing. So what it is, it's basically where there's an insertion or a deletion of the bases in the DNA of an organism which causes a genetic defect or a genetic condition out the other end. So it's where they use these things called indels as a form of gene marker. So you can see the top here, this top line could be a normal animal, so it doesn't have any form of a genetic condition, let's say. So we'll say that's the normal form of the animal's genome. Then you might have an animal down the bottom here that's actually got a 3 base pair deletion. So you can see it's actually missing that AAA at the top animal done. So it's actually got a deletion effect. Or on the other side, the genetic condition might be caused by an insertion. So you can see this animal has actually got 4 base pairs inserted as well, which could cause a genetic condition. So an indel is a form of gene marker that we can look at as well. So basically we've got SNPs, microsatellites and indels, which form the basis of all our DNA technology and our testing. Again, you don't need to know much more than that, I think, from an applied point of view, but you do need to be aware there's several forms of gene markers to utilise in this particular space. All right, so that's all the DNA theory I'm going to do tonight, which you're probably happy about. But I think that gives us a good starting point. And Gemma, I might just hand back over to you and see if there's any questions that have come on stream so far about what we've just been through. And Alex can also comment on these if she likes as well.
We haven't actually had any questions just yet, but if Alex would like to comment on it, that would be...
Yeah, no, you've done well. Keep going.
All right. Well, if that's the case, we'll keep moving on. But I would encourage people to, add their comments as we go along. Hopefully, I haven't bamboozled you, and you fully understand what I've just been through. So we'll now get on to the more applied topics. So what I want to do now is talk about the utilisation of DNA parentage and uses of DNA in Seedstock herds, and starting with parent verification. So I guess the reason for looking at parent verification is because you need to think about how accurate are our pedigrees in a Seedstock operation. Obviously, pedigree in a way is the currency of Seedstock operations. It's pretty important things to get right, because in a way you're trading in a pedigree of an animal. and it's utilised a lot of places, that pedigree, not just to put on a nice certificate and put online for people to see, but also in genetic evaluations and so on. It really has a lot of weighting. So I guess you have to think about, from a Seedstock producer's point of view, any way I can make the information more accurate, weighing up the cost of things, of course. It's got to be a benefit to your business. So I guess you've got to think about how accurate are my pedigrees, and we can always know there's going to be a level of error in this sort of things. It's just a human error that can creep in. It's not saying that any sinister things going on or anything, but basically there can be some areas that there's some error can creep in for pedigrees. And things like errors while you're AI, you've got the incorrect semen provided, you mix up straws accidentally, human error in recording which straw went into which cow, incorrectly assigning backup bulls versus the AI sire if you join them quite quickly one after the other. Things like determining sires for multiple sire matings, that's a pretty important area, but pretty hard to get the single sire sorted out if you don't do any parent verification. You also got on the cow side of things like mothering up, twins and heifers, mismothering happening, cross-mothering happening. You also got just standard management issues, bulls jumping fences and so on, the neighbour's bull or bulls swapping single sire mating paddocks. And you've also got bookkeeping errors that creep in. So there's no doubt some areas that can creep in to give us incorrect pedigrees, but it's where DNA's a place where it can improve our accuracy in these areas. I'm glad Alex is online because these stats are from him based on their Limousin progeny test example, where they've done a structured progeny test, and you would say that they've followed best practice to try and get everything right, but we still picked up some error in pedigree, because what they did is they obviously allocated sires and dams, but then they did the parent verification to see to make sure that's 100% right, because from a society progeny test you want to make sure the information's 100% or as close to as possible. So basically you can see in this progeny test program there was 300 mixed breed cows. They used 10 sires by AI and 3 backup bulls, and they did the DNA sire verification through the University of Queensland to check parentage. So it wasn't just sire verification, it must have been dam as well. Sorry, it was just the sire. So what they worked out is they worked out actually 12 out of the 227 calves out of this progeny test had incorrect sires recorded. So that's 5%. So as I said, they basically followed best practice, but you still end up with some 5% error creeping in. It's probably not a bad figure to think about from most Seedstock operations that are doing everything right. You could end up with between 5% and 10% errors in your pedigree, which can affect things if you don't do any sire verification anyway. So as you can see there, two calves swapped mothers. There was AI sire incorrectly recorded for 3 calves, 4 calves incorrectly assigned to a backup bull, and 4 bookkeeping errors for 3 calves. So that's where the error came from. Again, not a major thing to be concerned about, but again, if we can make things more accurate, then that has benefit spin-offs. So what are the benefits of parentage verification? Traditionally, we did parentage testing via blood typing, and I guess that was up to the early 2000s from what I could understand. That was a bit before my time, but I think it was up until that stage. And it's now replaced with DNA-based parent verification. And that can be either microsatellite based or SNP based, as I said at the start. We can now use either of those DNA markers to do our parent verification. But you can't cross over between the two, so you can't have microsatellite on your sires and SNPs on your calf and expect parent verification. It has to be one or the other, and that's why we're currently in a situation we have breed societies deciding whether they go across to SNP-based parent verification compared to microsatellites. And some breeds have transitioned, like Angus and Charolais. Some breeds are sticking with microsatellites until further down the track. The microsatellite panel depends on the lab, but I know that most labs work on a 2021 microsat panel to do their parent verification. You get very good accuracy with that sort of things. From a SNP point of view, it's where they're looking at around 100 SNPs at the moment. I know the Angus, I think through Animal Genetics Lab in University of Queensland, have 96 markers, and they're the standard, what's called the ISAG, the internationally recognised markers. They use that panel. At Pfizer, I think they've got 110 in their panel, so they use a slightly larger panel. Or what the other good thing is about SNPs in a way is if you do a larger profile, so say you do a 10K test or a 50K test, if you're an Angus breeder doing your 50K performance panel, then basically you can pull those SNPs out of that as well for parent verification, as I understand it. So that's information that we're going forward with. Another bit of information that I understand from the University of Queensland in any case, they're actually looking quite soon, by September, to go to a slightly larger panel, so 150 to 180 SNPs. They're just doing some trialling at the moment. So they're looking to go from 96 to 150 to get, I guess, a higher reliability in their parent verification for across the majority of breeds. So they're currently transitioning to that, and it'll be interesting to see how many breeds transition after that point. So basically, parent verification is a tool that can be used to reduce pedigree errors, remove the requirements to mother up animals, enable multiple sire matings, and obviously still get a single sire outcome for your pedigrees, and obviously increase the accuracy of pretty important things such as genetic evaluation results, inbreeding coefficients, estimates, and GPROM genetic condition analyses. So without accurate pedigree, those bottom three things can be really thrown out of whack. With accurate pedigrees, then that information becomes more accurate as such. So DNA technology can be utilised to really get those accurate pedigrees in. So some considerations in regards to parent verification, and again I'll just say that I'm moving through here pretty quick and there's probably a lot more around parent verification, but this is just some general comments because we've got a lot to cover tonight. Some considerations: breed societies generally have regulations in regards to DNA profiling parent verification, and you should all know what they are for your breed. Some breed societies like Wagyus are quite high level to get in their herd book, as you actually need parent verification to sire and dam to get in. Some breeds just need a DNA profile on file for a sire or an AI sire, but they mightn't do any parent verification. Down to the, I guess, to the level down, which is basically in regulation where they say we can do spot checks on DNA to get a parent verification there. So it really depends on the breed, and check up with your breed if you're not sure. Breed societies will generally have contracts with one or more of the labs, such as UQ or Zetas, which is the Pfizer equivalent, or old Pfizer, I guess you would say. And you should know that for your own breed, but chalk that up. The cost varies depending on the type of DNA sample, the number of samples, the type of profile, so whether it's a SNP or a microsat, and whether parentage verification is required. The indicative cost is around $15, and from what I've seen, particularly what I've learnt from how the Angus operate, who's transitioned to SNPs, they're getting the profiling and parent verification done for around $15 to $20, might be even around that $15 mark, I think. And most microsats tests are around that $22, $24, I think. So that's the situation for the indicative cost, but please check with your breed society to get a better cost indication if you're not aware already for your own particular breed. All right, that's a really quick snapshot on parent verification and some thoughts there, and also I guess a little bit of a look into the future about where it's going to do parent verification with SNPs. I'll just hand back now over to Gemma and ask is any questions coming from either of the first two sessions.
Yeah, Christian, so I've got a few questions coming through. So with the SNP panel,
Good.
is it possible to do more testing? So if they do a parentage verification, could they also test for disease statuses, etc.?
I understand you can actually, when you submit a sample, you can say you want a number of things done. However, they won't use exactly the same panel, as I understand. Alex might comment, exactly the same panel. Like, say, for example, they use the 96 ISAT marker. That same panel won't be used to do the genetic condition testing, but they will still bundle it in and say, if you tick these boxes, you can get that testing done based on this sample submitted. What's your knowledge of that, Alex?
Well, the first thing is that if you send a single sample of DNA in, you can get a number of tests done. For example, you could get a DNA type, you could get a horn pole, you could get a colour, you could get etc. We were told that Identity, GeneSeek, with their 10K chip, would be able to give us pretty much everything, including the SNP panel for parentage, horn pole, colour, and maybe and the disease traits. There's certainly enough SNPs on that panel to do it, but right now I don't think we are being delivered the whole package. We send samples over there and we get a 10K SNP back very quickly, but it's a bit more delayed for horn pole, colour, disease. But technically there's no reason why you can't get all of those results out of one 10K SNP test.
Right. Thanks, Alex. Is there another question? Gemma, we might take one more before we move on for time.
Yeah, just one more question. Is it expected that in the future parentage verification testing will come down in cost?
Well, from what I've seen, and again this is looking at the Angus example who have transitioned into SNP-based profiling, they're getting basically through their SNP testing, they're around that $15 mark. So I guess you'd say traditionally microsatellite testing was around the $22 mark, $24 mark. So I guess you would say just on that alone, you would say that transitioning into this newer technology, you may get some economic benefits out of it. But I think where you'll see most of the benefits is, sort of fits in with the last question, is where they'll be able to bundle things together. So you won't just get a profile for parent verification done. You'll actually get a number of tests bundling together, whereas where you'll get your economies of scale in a way, I guess you would say. Do you have a comment on that, Alex?
Yeah, no, that's correct. If you just do basic parentage with SNPs, you can get it down to $15, but the advantages are going to be in bundling. So I agree.
Okay.
Thanks. All right, we might move on for time, Gemma, but please keep your questions coming in, and we'll make sure we pull up for a couple of other times to handle those. Okay, the next section I want to cover is on our genetic conditions. So we've been through theory and parent verification. I now just want to go through genetic conditions because this is an important part for DNA technology. And I guess we need to describe what genetic conditions are. A genetic condition is a disease that's caused by an abnormality in an animal's DNA. So it's where we can think back to that indel. So we've got a deletion or an insertion in the base pairs in the sequence. So that's normally where we get our issues. And abnormalities can range from a small mutation in a single gene to the additional subtraction of an entire chromosome or set of chromosomes. And they can lead to a range of symptoms. So they can lead to things that are quite subtle in effect, to things that are quite not subtle, such as being a lethal, the calf aborts or dies early after birth. So it can range in effects as such. For most of the genetic conditions we look at what cause recessive conditions, and they're generally, the frequency of the carrier animals are usually quite low in a population, and the expressions of the conditions are quite rare. So you wouldn't expect to see hundreds of animals out in the field with these genetic conditions. You'd see the odd one here and there, but it's where we must make sure that we don't multiply the problem to get a bigger problem down the track. The risk is obviously increased by inbreeding or reducing our genetic diversity in a breed, so it's where we can get these carrier animals for genetic conditions being stacked one on top of each other, and that's where we can run into problems. So if we can think about inbreeding genetic diversity as well and trying to maintain our genetic diversity, can assist in this. But there's obviously a range of tools that can help assist in this as well, which I'll go through, such as DNA. And there's a large number of genetic conditions that exist across all breeds and species. So genetic conditions and genetic defects is another way of putting it, is not something that's just unique to one breed. Most breeds have got at least one. There's probably a lot of unrecognised ones as well. This next slide really shows the incidence of genetic conditions across species, but also in more details in the bovine species in the red. So basically what it's saying is if we just look at the cow side of things, there's 405 recognised genetic conditions. 115 of those have been identified as being under the influence of a single gene or a single locus, and 68 of those have been characterised by a DNA test. So there's quite a lot out there that we know are characterised and we can do DNA testing for. And there's actually 136 of those 405 that also have what we call human equivalents. So it's a similar genetic condition in humans, so it sort of spreads across species in a way, but similar in effect. And you can see the differences. So we just need to get to the point that these things are around, they're going to continue, and we'll find more of them, so we just need to work out how to manage them, and DNA is one way we can do that. Most of the genetic conditions we're aware of and are identified in the beef world are what's called recessives. So all that basically means is we need two copies of the recessive gene to come together to see the animal being affected. Okay, if an animal's only carrying one copy of the recessive gene and one copy of the normal gene, then the animal would be normal himself. It would be determined a carrier animal. If the animal's copying obviously two copies of the normal gene, then there'd be no issues there. The animal would be free, wouldn't pass any of that on, wouldn't be a carrier. And you can see that in the mating combinations. So I've just highlighted what the gene would be. It would be a big N would be a free animal, a big N, little n would be a carrier, and two little n's would be affected. So I'll just pick those letters just to utilise in this example. So you can see the first one in total green on the bottom left-hand side. We're joining a free cow with a free bull. We'd expect 100% of the progeny to be free of that genetic condition. If we joined a carrier bull, so they're carrying one copy of the affected gene and one normal copy, and joined to a free cow, we'd actually get 50% of the calves would be free, but 50% of the calves would be carriers. We wouldn't actually see the symptom because we don't have two copies of those recessive genes coming together, but you'd still potentially breed on that problem or potential problem. And in the case on the far right hand side, we've got the carrier bull joined a carrier cow. 25% of the time the calf would be free, so wouldn't be a carrier or wouldn't be affected. 50% of the time we'd get a carrier animal, and 25% of the time the calf would be affected themselves. So they'd see the symptoms, and that's where you might come up and say, what's going on here? What's the symptoms? What's this issue? Is it a genetic thing or is it something else? So that's the somatic inheritance we're looking at for most of our recessive genetic conditions in the beef industry. Tools available to manage genetic conditions. The first one is we do have DNA tests available for numerous conditions. For example, Angus have 3, AM, NH and CA. I won't go into these in more detail. Please, if you've got any questions about your own breed, get in touch with your technical officer. Wagyus have got 4 recognised ones. Herefords, I think I've just got 3 that have come online. Limousin have got 1 that I'm aware of. Brahmans have got 1. So there's a range of breeds out there with DNA tests and genetic conditions that can be managed through this particular process. As with parent verification, the DNA testing can generally be submitted direct via the Breed Society. I think you can deal directly with a lab too if you like, but mostly via the Breed Society is recommended. And please check your price with your Breed Society or lab. The price ranges between $20 to $50 per animal. So if you're wondering if an animal is a carrier type animal, then you can get it tested and know for quite a low cost, depending on what particular genetic condition you're looking for. The other add-on to this I just wanted to mention tonight is GeneProb. So it's where we can get real bang for our buck in our DNA testing in the Seedstock sector for those breeds that have applied it. GeneProb software that basically was developed by Brian Kinghorn at the University of England, a great quantitative geneticist. And basically what it does is analyse the pedigree and calculates the probability of an animal, or of an untested animal, being a carrier. Because it knows, obviously, if the DNA results have been recorded with the Breed Society, we have it recorded on the Breed Society database, the GeneProb analysis works up and down the pedigree and says, okay, we know these animals are carriers, or they're affected, or they're not affected, so we can then work out probability-wise what the progeny may be, or what the particular animal may be up and down the pedigree that isn't tested themselves. The results are presented online on Internet Solutions, which I'll show you in a second, and they're updated regularly depending on the breed, but you know, at least once a month you get updated results. These are the way genetic condition results may be displayed. Some breeds mightn't actually run GeneProb, I should say. They might just display the genetic condition if they've been DNA tested, and whether they're free or carriers or affected. But some breeds have got GeneProb to add on to that. So you can see here, this is just looking at a condition called AM, which I think we have in Angus and also analysed in Murray Grey. And you can see the top animal here is AMF, so it's tested free by DNA from the lab. The bottom animal is also free. It's saying it's free, but it's untested, so not tested based on pedigree, expected to be free. Obviously, we're not really 100% sure if the animal's free until it's actually DNA tested. What the bottom one is estimated off what we know about the animal's pedigree. The next two down are ones when an animal could be a carrier. So the AMC is it's tested by DNA, tested a carrier for that genetic condition. The one up the top is basically looking at a probability. So for AM, for example, if you had 25% in there, it would say, based on the pedigree, we know that that animal has a 25% chance of being a carrier. Could range from 50% down to 1%. Okay, so there's an estimate there on the animal being a carrier as such. And obviously, if an animal's affected, then you would pick that up as well as being an AMA. So basically, the first two is just the code for the genetic condition. In this case, we're using AM, and the next few are basically just saying whether it's tested for a DNA and what the outcome is, or whether it's a GeneProb analysis. This next slide then just shows an outcomes from the Angus situation. So we can see here I've got some sires there. There's some of the published sires. And I've got highlighted there, you can see up the top we've got animals that are DNA tested free, and it's highlighted in green, which is a nice way to look at it. So you can see these top animals free by DNA test for AM, NH, CA and MA, which are 4 ones you can DNA test for in Angus. This one down here, AJC A83, is free of AM, but he's a carrier for NH by DNA test. And you can also see he's free of CA, but he's free but untested. Down the bottom here we've got some other animals. You can see this top one here, Siegfried, Black Mr. Siegfried. He's 25% chance of being AM carrier, 25% chance of NH, and he's free untested for CA. So that just gives you some ideas of the how the results are displayed. Now, does this technology work to actually reduce the incidence, both the DNA testing and the gene prob, to reduce the incidence of carrier animals in the population? I think there's no doubt by saying I think any breed can replicate this if they had the stats. As soon as you get a DNA test released and it's normally uptake pretty quickly by the breeders, and we see a reduction in the population of carrier animals, that's for sure. So basically this is just looking at the Angus frequency for AM, NH and CA, which are the recognised genetic conditions. You can see back in 2008, 2009 we had the AM and NH test developed, the DNA test, and NH was sitting around 8% of the population was a carrier for this particular genetic condition. And you can see it's actually dropped back quite significantly. So you can see now down today with the DNA testing and the way people are bred, the carrier level is now down around 3% or 2 to 3%. AM's gone the same way, and CA, which the DNA test was implemented a lot later, has gone the same way as well. So we're down around 2% of the population. So DNA tests are definitely utilised and can be used to decrease the populations of carriers in the population, as shown by this Angus example. All right, that's a really quick run through on genetic conditions, Gemma. So we might pull up again for probably just one question this time, if you've got time to hand that question over.
Yeah, sure, Christian. If someone was to do some DNA testing within their herd for genetic conditions and they recognised that a few animals were carriers or affected, how would they go about managing that within their herd?
Oh, right. Yeah, good question. I think the main thing is to work out what animals is it. Are they sires? Are they cows? The first thing to probably talk about is don't be overzealous with. I think a lot of people in the past got caught up and maybe thought I need to eradicate this out as quickly as possible. But you need to think about managing it in the longer term. How do I get around it? So I guess you'd want to go into a herd by herd on detail, but my recommendation was basically don't utilise any sires that are carriers for that genetic condition going forward. For your cows that have been identified as being a genetic condition carrier for that, you can still keep those in your herd, breeding herd, particularly if they're good high productive cows and high performance cows. But you need to probably join them with non-carrier sires and probably also do some testing on their progeny to see whether they may have carriers in their progeny, so you don't want to breed those on yourself or sell those off potentially without being fully transparent in the information being supplied, of course. So it really depends on the individual situation. That's sort of the general thoughts I've got at the moment. Alex, have you got anything to add to that?
Yeah, no, that's a fair comment. I mean, as long as you join a carrier cow to a clean bull, you're going to get 50% of clean progeny, but you are going to have to test to work out which ones are clean and which ones are carriers.
The other thing is you can, if your breed's running a gene prob analysis, you can do a bit of testing and then you can work out probability-wise where you might have to focus your testing. So if your breed's got that technology available, it can really help you hone in on where the testing might have to be done to even further fast-track getting it out of your herd without having to go over the top and just eradicate. Okay, we might have time for one more question. Gemma, if you like.
Yeah, so just one more question. Is there a recommended level of carrier status before you should start looking at DNA testing your animals? So if you've got 5% of your herd or, 10% or something like that?
It's a pretty difficult one to answer. Alex?
No, I think it's easy. I mean, if you have a disease, if you have a genetic condition in your breed, then the first step is to make sure you test every sire and make sure you only use clean sires. And then it's an individual herd decision whether they bother to test their cows. But I can tell you that in our breed we had about 10% of carriers of proto. And simply by testing the sires and making sure that people and people only using clean sires, that carrier level's down to 1%.
Okay, thanks, Christian.
Thanks, Alex. Yeah, thanks. So do you think they'll, when you get down, and I had this question the other day at a field day, when you get down to that 1% level, Alex, is there any point, like, if it stays at 1%, is that acceptable? Or do you think you should try and drive down to 0%?
No, I think by my calculation, if you've got a 1% frequency of carriers, you only get something like chance of 1 in 4,000 actual diseased animals. Yep. However, in our experience where we import bulls from overseas who don't test as rigorously as we do, we will continue always to test certainly AI size.
Great. Thanks, Alex. We might move on, Gemma, for time, but we'll come back to more questions in a second. Okay, the next section I want to go through is qualitative traits. So when we talk about qualitative traits, what we're really talking about is the traits that there is a sharp distinction between phenotypes, and where there's only generally one or a few genes are involved in the expression of the trait. So you can think of coat colour or poleness in this regard, and that's what I want to focus on now. The genetics for black and red coat colour, and apologies for the typo there, this is the first one we'll comment on because there is a DNA test out for it. There's 3 possible alleles or alternative forms of the gene, I guess you would say. There's the black allele, the red allele, and the wild type, which is thrown in just to complicate things a little bit, but not too much. But what we're really looking for here, I guess people, depends on which side of the fence you sit in, on what breed you're involved in, what you're looking for. Some breeders might be looking for what's called homozygous black bulls, so that means we can make sure that all the progeny are black, or you might be a breed of a red breed, and you might be after a red carrier, so that the animals are black itself, but you might be after the red gene to go into your breeding program to read some red progeny and open up your gene pool a bit potentially, like if you're a Red Angus breeder looking in the black gene pool, for example. So it's basically black is the dominant over the 2 other alleles, so black's dominant over the recessive red or the wild type particular genetic form of this allele. So basically what that means is the top animals, as you would see here, will all come out black in phenotype. So EE, if we got 2 of those together, because it's dominant, you'd end up with what's called a homozygous black animal. You'd expect 100% of the progeny to be black. If you ended up with an EA in a particular animal, the animal will be black itself. It's carrying 1 copy of the black allele and 1 copy of the red allele, and that means it would be a carrier for red. In this case here, for the wild type, it would carry 1 copy of the black allele and 1 copy of the wild type, but the animal would still be black itself. The red animals, for example, this animal here would be red. It's got 2 copies of the recessive red, so you'd get the red animal coming out. Or if it's got 1 copy of the red and 1 copy of the wild type, the animal will be red as well. The other here, if we get 2 of those wild type alleles together, it could be any colour combination, and they're very rare to see, but it's still something you can throw a little complexity in the mix for the black-red coat colour. But that really gives you an idea of what the DNA test results might look like. Historically, the only way of identifying the genotypes and the resulting phenotypes was through progeny testing, but now there's obviously a DNA test available. It's been around for some time. It looks at this, what's called the extension locus, and what it does is differentiate between homozygous and heterozygous black animals. This test is normally available via the Breed Society from those 2 labs, University of Queensland or Zetas. University of Queensland, I think, just call it dominant black and recessive red. Zetas call it GeneStar black. And the indicative cost is $15 to $30, but you need to talk to your Breed Society or the labs directly about the specifics there. But basically, it's a test that can be done to pick up homozygous black and heterozygous red, or in that particular vein, and how you want to use that's really up to you. In some breeds, you might get a benefit in having sires that are homozygous black, for example, if you're selling them to herds and want 100% of the progeny to be black. So that's one way to look at it, and that's a DNA test for a qualitative trait. The next one I want to just go through now quickly is the genetics of Paulus. This is another qualitative trait. So obviously there's a DNA test out there now for this particular thing. It's not as simple as we once thought of saying, you know, if an animal's horned, it's horned, if it's polled, and something in the middle. But it's where if we can really fast track herds to breeding polled, and I think that's where most industry is going now, to breed pollness, if we can find those homozygous polled bulls. So it's basically where there's several genes involved. There's the polled gene, which is on chromosome 1, and that's what we're going to be focusing on in this talk. There's also SCRS, which throws in a mix, particularly in tropical breeds and other breeds, and that's located on a different chromosome. And there's also the African form gene, which is, I've got some question marks there. It's something that's talked about a lot, but probably not a lot of information on, which can really throw, if it's there, could throw some complexity into the mix in regards to tropical cattle. So basically when we're talking about the polled, which is what we're going to focus on tonight, is we're looking at, for example, this animal here carries the two forms of the dominant form of the allele, PP, and the middle is the would be a polled animal itself, but it's actually a carrier for horn, so it's polled and horned. And the animal here itself, on the right-hand side, is a horned animal, and you can see it's expressed in the phenotype. So what we're really trying to identify as an industry, I think, are these animals that are PP, so that means 100% of the progeny will be polled phenotypically, which is probably a good thing for welfare and all those things going forward. SCRS, just to add a bit more about that, are sex-dependent and are only expressed in the polled animal. So you don't get SCRS expressing in horned, it's only in polled, and only if that particular allele exists on chromosome 19 and it's sex-dependent. But we're not going to go into any more details about SCRS tonight, really focus on the horned polled. About the DNA test for pollness, the precise location of the polled gene has not been identified. We should say that. We're basically using DNA markers, which are evaluated genotype at a linked gene marker. So we're actually using it to give our best estimate of the animal's homozygous polled or heterozygous polled, etc. And we don't have a gene marker for SCRS at this stage, so we're really only focusing on that pollness. Companies offering the test at the moment are the University of Queensland, Pfizer, and I should say that Zeta, so I didn't update my slides there. But there's also overseas labs offering this DNA technology. Alex has always mentioned one in the States, Igenity, and they also offer this particular pollness test. It's different to the beef CRC one, and I'll get him to comment about it when I pull up in a second. Basically, the labs in Australia offer the beef CRC developed test. It's derived from the beef CRC, but it's currently only suitable for Brahman, Santa, Gertrudis, Droughtmaster, Hereford, and Simmental. And it's due to there being this ambiguous allele effect, we can really throw out the results in some other breeds and doesn't give us enough returns that we're highly confident in. So it's where if you're a Brahman, Santa, Gertrudis, Droughtmaster, Hereford, or Simmental breeder, you can actually utilise this test as it stands now. For other breeds, a bit more work needs to be done. It's really most suited to Brahmins, you should say, because that's where the test was developed on a Brahmin population, so you'd expect that to be the case. The price might have a small variation in there for $20 to $40, but please talk to your breed society or your labs to get more details about it if you're interested in going down that way. So how are the the results reported for this particular pollness test? Remember, we're dealing with a gene marker which is linked to pollness. We're actually looking at the direct gene itself, or genes itself. So what we've got here is we've got a test that generates probability of each genotype. It's not 100% definitive. So you can see here, for example, the results might say we are 91% confident this animal's homozygous polled, 8% it's recessive, so polled, carrying one copy of the polled gene, one copy of the horned, or homozygous polled, so it's 1% chance of it being horned. You'd probably know by the phenotype if it's that, but if animals have been dehorned correctly or the right way, you may not be able to pick it up if you bought the bull as such. But basically that's the way the results, it's got a probability estimate around it, which is important to understand. It's not 100% definitive, but it's still fairly high. So you can see the way some people are displaying results. This is out of a Hereford breeders catalogue that I know. You can see here animals that are PPs. He's basically saying there's 90% chance of the animal being homozygous, being true polled, with 90% likelihood. If it's 5%, if it's pH, there's a 5% likelihood. If it's 48%, then there's 48% chance there. So they give you a probability estimate to go with that, which you need to be aware of. So that's just a summary of our quantitative traits. So we've been through black, red, and we've been through horn pole, just to give you more details. Again, there's a lot more information about this out there, including we've got a webinar clip specifically on our horn pole test from the beef CRC last year you can look at. But is there any questions, Gemma, we can go through before we move on?
Yeah, I've just got one question for you, Christian. If you've got an animal that is DNA tested as homozygous polled, but you've been getting some horned calves from it, what would the next step be?
Yeah, good question. Alex, you've probably come across that a bit in your breed, have you?
If you're starting to get horned calves from a homozygous polled bull, you can assume the test was wrong. What I should also add is that we are very close to getting a much better Australian test. The initial test was one marker, and in Limousin experience we get a lot of ambiguous results. I think we're just a month away from moving to an 8-marker test, which will probably give you at least 90% accuracy. So we're going to move away from a situation of getting 45%, 48%, 25% and 5%. We'll be like the identity test. We'll either get a result which says yes, 90% or better chance of homopolled, or it's not homopolled.
Yep. So it's getting rid of that ambiguous side of things, which is what. Yep.
By using 8 markers, and the identity test, which we've used for some time, I think's got 18 markers, but from what I hear, the 8-marker new Australian test will be probably just as accurate as the 18-marker identity test.
Have you heard of a timeline on that, Alex, at all? When that may be available, or is that they're still working on it?
We're currently having some animals that were ambiguous being tested right now, and I believe by the end of August it'll be implemented.
Great. Okay. We'll keep our ear to the ground about that one, because I think that'll be of real benefit to the technology, and we'll make it applicable across more breeds as well, which I think is a great thing. Okay, Gemma, we might move on for time. It's getting away on us, so again, keep your questions coming in, and we'll answer some of those at the end. Okay, the next part of this presentation is going on to the 4th area, and this is really looking at DNA for production traits, and how particularly you can have results available now, and how that will be can flow into the breed plan evaluation. But basically the concept of DNA testing for production traits is basically outlined here. Basically what happens is they're developed in a reference population, so we have a group of animals that are recorded for a range of traits, or maybe just one specific trait you're really interested in, but they're recorded for something really well. They've also got DNA information and normally SNP profiles, maybe ranging from a 7K profile to an 800K profile, depending on what we're looking at. And it's basically where the DNA test is developed in this reference population. And then what we do, so we basically work at the link between the SNP, the sequence of SNPs, and the performance of animals. So you might say we know that the animals that are higher marbling performance, for example, will have this sort of SNP profile compared to animals that might have poorer calving ease, have this sort of SNP profile. So that's where it's looking at a reference population to do that. And then what we can do is we actually test animals outside that reference population, but they should still be related. So it's not where you'd have a Brahman population and test an Angus. It's where you'd have an Angus population up here, and then you test another Angus animal that still had some relationship to this through pedigree or whatever. So a current Angus, for example, or a current Brahman and a recurrent Brahman population up here. And from there, we use this in-quarter prediction equation, which is the fancy geneticist word, to calculate the molecular breeding values based on a SNP profile. So it's really generated, and all the work's done up in this reference population, and then we just take a DNA sample of this animal on a SNP profile, such as a 10K or a 50K, 50,000 SNP platform, and from there we can get molecular breeding values, which are basically just EBVs based on genomic or DNA information only. So just to go through some considerations with this, an MBV, or the molecular breeding value, I should say they're called a range of things, these MBVs. You might say from Pfizer we've got MVPs, or you've got, I've thrown around the other day, was DGVs, direct genomic values. They're all referring to the same thing. EBVs generated from DNA information only, which is based somewhere off a reference population. And the other thing you've got to understand is accuracy of these things aren't 100% because we're dealing, they're basically an estimated breeding value, we're dealing with accuracy in the same vein. For most of these traits, particularly the ones going through breed plan, there's not a few genes that have a major effect. Most of these things have most of the SNPs in a large profile might have a small effect which add up to some sort of effect down the track. The more SNPs, the greater the proportion of the genetic variation and accuracy that will be explained potentially. So generally speaking, the bigger the profiles and the better the reference population, I should say as well, the more accuracy we get in these particular tests. The reference population should be of sufficient size, as I've said. You need quite large reference populations to get accuracy in these tests, as we're working out quite readily through the beef industry now. And that's easier said than done, because you know if you look at a trait that's hard to measure like net feed intake, it actually takes a lot of time, a lot of money to get those traits together in a reference population. You still need the phenotypes to do the referencing off. So it's something where we need to build on in the industry all the time. Also, animals being tested must be related to the animals in the reference population, as we've already talked about, and that's why we've got breed-specific tests. And they must also be updated regularly, as in you wouldn't be able to develop a test today and then expect it to work even in the same population of cattle in 20 years' time, because the population moves, things change. So it's where you need to update the particular prediction equation, I guess you would call it, over time. One thing I think we've definitely worked out as an industry, and hopefully we don't have to say this too often, is the most beneficial application for this DNA technology at this point in time is to flow through the breed plan evaluation. It's really just an additional form of information. It's like if we've got some additional pedigree or a different performance measure, some scanning or something, we've actually got DNA test information that can be utilised in breed plan. And that's where it can be of real benefit. So it's where we're combining it with our pedigrees, our phenotypes. We're combining DNA to give us breed plan EBVs still out the other end. But I guess what's the benefit of using DNA technology in the breed plan evaluation, or the potential benefits? What it can do is it can really add extra accuracy to existing breed plan EBVs. That's really the basis of it. If it doesn't give us any extra accuracy, then what's the point? That's where it has to really be focused towards. The good thing about DNA, though, is it can be collected early in an animal's life, as in you don't have to wait until an animal's 2 years old to collect it. You can wait until it's quite young and get a DNA profile back quite quickly, which can give us accuracy on a young animal, which can be real beneficial compared to, say, a trait that mightn't be able to be recorded till much later in life, like, for example, some of our maternal type traits or some of our later carcass traits. So there's some of the things we can utilise DNA to get some good accuracy early in life, potentially. They offer an alternative for traits that are costly to measure, like I mentioned already, net feed intake. They still need to measure it somewhere because we still need a reference population, but it might mean that the rest of the industry doesn't have to measure it to get the phenotypes for it. They are a direct measurement of animals' genetics, that's for sure, but as I said, they don't have 100% accuracy. And I think they're also a very viable option for small herds. For example, breed plan in phenotypes work on contemporary groups, so you need animals that are got head-on-head comparisons to get the usefulness of their phenotypes. The DNA technology doesn't work that way to create that sort of information. Basically, you might have an animal that's been sick, or there might be something wrong with it, or it's in the same single animal contemporary group, and therefore its phenotypes aren't going to be much use, so you might do a DNA test to build in some accuracy for that particular animal. So that's a way to think about it as well, and I've seen that being done already. So what DNA tests are available for production traits? I'll just go through them now quickly. We've got Zetas have the first two there. We've got the first one is the 55 SNP panel, so there's only 55 SNPs in that. And it's basically where you can get results back for gene star tenderness, gene star marbling, and gene star feed efficiency. The gene star tenderness is probably the most important part because that's the part that can currently flow through into our trial shear force EBVs for Brahman. Moving on, I guess the one that's you probably see more about in industry these days is the Pfizer Animal Genetics or Zetas Angus HD 50K panel. I think actually it's got 54,000 SNPs in it from what I understand, but we call it the 50K panel. And basically it gives us 16 molecular breeding values out of that, so ranging from birth weight down to net feed intake. You can see them all there. and a good wedge of those goes in the breed plan, which I'll talk about in a second. But we also have other breeds out there with genetic specific tests. We've got prescribed genetics, and by UQ or Pfizer or Zetas, I understand, do this as well. They have a few there that are related to marbling and the quality of fat. University of Queensland are also offering the myostatin F94L, and Alex might like to comment on that in a second, which gives us some differences in yield, I understand it, maybe some other traits, without looking at that really extreme double muscling type thing. And Pfizer also do a Piedmontese myostatin type gene. So there's a range of tests available which impact on the production traits. And there's also a range of tests available for overseas labels like Igenity, who offer additional things, which I won't go into now, but you need to be aware of those. But we do have labs in Australia that offer this particular information. So what information is included in the breed plan? Because we've said this is basically the best way for it to be incorporated for a breeding decision. As I've said up the top there, and I've said already, the Pfizer GeneStar tenderness MVPs from the 55 SNP product is included in the Brahman Shear Force EBVs, and that was incorporated quite some time ago in October 2008, so it's not a new thing. It's been done for quite some time. The second point there is for Angus, based on the Angus 50K Zetas product. 12 out of the 16 traits that are currently we get MVPs for, or molecular breeding values for Pfizer, are blended into the Angus group breed plan analysis, and they're the traits there. So we actually blend in calving age direct, calving age daughters, birth weight, weaning weight, yearling weight, mature cow weight, milk, carcass weight, rib fat, eye muscle area, intramuscular fat, and scrotal size. So that's additional information that goes into the breed plan analysis for Angus based on this 50K panel and the reference population that's been worked out on. So what value is of this information to increase EBV accuracy? Because that's really the point we were aiming for. And this slide just shows it nicely. So what it's basically saying down here, if we've got an animal, and this is existing EBV accuracy, if he then had these MVPs added, what would the accuracy actually go up to? So, and this is for the carcass rib fat EBV, and this is probably, just to give you an idea, most of the accuracies we're getting out of the MVP from the Pfizer panel, the Zetas panel, to go into the breed plan analysis range is anywhere between 10 to 40% accuracy based on an EBV accuracy. So you see here, if you had an animal that didn't have any accuracy, you didn't know anything about it, for rib fat EBV, you'd actually jump up quite a lot, 40% accuracy. I guess most animals are in Angus, or most other breeds we know something about. So if the animal had just had his own rib fat scan and didn't have any pedigree, which would be a bit unusual, but it could be the case, then the accuracy would already be, for that animal, would already be about 35 or 35 odd percent. But with the MVP, it would go up to about 50%. So it's still giving a reasonable bump up in accuracy there, particularly on a young animal we not know much about, or an animal we just don't know much about at all. If you then just look at a mid-parent, so we'll say this animal, based on his current EBV analysis, had 75% accuracy in his sire and 60% in his dam, he might end up with about 55% accuracy. If you add then in the MVP on top of that, then you would actually get the animal up to a bit over 60% accuracy. Then obviously if you had the animal's own scan and his, sorry, down here I was looking at, if you had the animal's own scan in the mid-parent, then the further you go up here and the more information we have, so lots of scan records on the sire or 15 carcass progeny, then the less benefit you're getting out of the MVP additional information. So the real benefits down this bottom end, and you can think of that of animals we don't know a lot about, or young animals where we haven't had time to record that information yet. So if we need to make some early selection decisions, it can give us a bump up in accuracy as such. So hopefully that helps describe about how the MVPs fit into EBV accuracy over time. Lots of value down here, not so much value up this end, where we already know a lot about the animals. This is just a display of how the MVP information is displayed on the online facility for Angus. So basically you can see if animals have been recorded for the HD 50K profile with Zetas, and that's been loaded in the Breed Society database, then you'll see that it gets this little trait indicator here. So you can see this animal's had a birth weight, 400-day weight, scrotal size, scans, etc. himself. He's got heaps of docility progeny. He's been using 42 herds. He's got 1,600, nearly 1,700 progeny analysed, 700 odd scans, 81 carcass progeny and 2 performance daughters. So you can see he's quite a high accuracy size, but he also got the Pfizer 50K product there. So for that particular animal, it's probably not adding a lot, maybe to some of his carcass traits, but probably for the other traits where he's got a lot of progeny recorded, it's not really giving you much extra that we don't already know from his phenotypic information. But you can see where it's displayed, and it is going into the breed plan evaluation. Okay, so that gives us, again, a quick overview of the particular production traits available, and how they're incorporated in the breed plan in a very quick term. So is there any questions, Gemma, you'd like to pass on?
Yeah, Christian, so we've probably got one question. If you were developing the DNA test for production traits in the reference population, if you had a reference population that used multiple breeds, so they were crossbreds, for example, would you be able to use that to create a prediction equation that went across multiple breeds?
I think the short answer to that is, based on their current knowledge, it would still give us less than optimal results, because if you got a reference population that has crossbreds or across a range of different breeds, and you're really applying it to a specific breed, then you're still going to get a better MBV or MVP analysis if the reference population is on that specific breed as such. So I think the further we get away from what the reference population is to the animals we're selecting on, the lower the value of the results as such. I think I'm interpreting that right. Do you want to add anything, Alex?
Yeah, no, you're pretty close, Christian. The only issue is if you don't have lots of information on individual breeds, if you just say took the Euro breeds, it may be that you'll get better predictions out of using a bigger database including those 3 breeds than the individual breed databases. And certainly Mike Goddard has always argued that he thought in the longer term we'll get better prediction equations using a combination of breeds. But you're right, the most accurate predictions have come from where an individual breed such as Angus has a relatively large database of Angus. So yeah, but it's not a clear cut.
Yeah, thanks, Alex. Just for time, Gemma, we might move on and just finish. I've only got 1 or 2 slides to go to look at the future, and then we can take a few more questions before we finish up. So I'll just move on now. But we'll definitely finish up with a few questions because I see there's a few coming in. Okay, so this is always the dangerous section, crystal balling, because this technology has changed quite dramatically over the past 5 years when I've been involved, that's for sure. So I guess what I'm saying here is, what does the future hold? What do we expect next for the DNA technology? And I've just got one slide with a few dot points on it which covers this area. So I'm interested to hear Alex's thoughts on this as well, to see what he thinks. But this covers across the gamut of the genomic technology we've been through this evening. I think one thing we will definitely see over the next 12 months is more breeds transitioning to SNP-based profiling and parent verification, and particularly when we get a larger SNP-based panel which may be useful to more breeds, like the 150 SNP panel through the University of Queensland. Then we will see that transitioning happening. Saying that, not all breeds will do it. There's some breeds that might stay on microsatellites longer, but over time I think we'll see that transitioning take place. The other thing which I'm glad Alex has also mentioned is we'll also see an increased accuracy of the horn pole tests for more breeds and less ambiguous results. I think that's really going to add benefit to that technology, DNA technology. One of the other main things we're going to see, and some breeds are doing this already, is we're actually going to be storing the SNP profiles on the Breed Society database. So what that means is when you actually send and this has to be arranged with the labs, I guess you would say, but this is the general concept. When you send a DNA sample to a lab and you get a SNP profile, whether it's 150 SNP or a 50,000 SNP panel, it's where you won't just get the DNA results back for the particular test you're doing, but you'd also ask via your Breed Society for the SNP profiles back. So you'd actually get, if you did a 50K panel, you'd actually get the SNP results for all those 50,000 points, and that would be stored on your Breed Society database for future utilisation, genetic evaluation and so on. So that's definitely where breed societies are transitioning, and the technology is being set up now for that to happen. I think one thing we'll also see to add into the incorporation into breed plan is at some stage we'll also see delivery of the beef CRC prediction equations, which has been talked about a lot but isn't actually being delivered through breed plan as of yet. But basically I think we're pretty close to the point where we will get a range of traits delivered for Angus. We'll also get a range of traits plus some important fertility traits being postpartum and estrus interval and age at first corpus luteum delivered for Brahman. Sangatruus and Shorthorn looks like we'll get some growth traits considered to go in to the breed plan evaluation on the DNA test. Whether that's valuable or not I don't know because it's a pretty well recorded trait anyway. For all the other breeds, it looks like the DNA test isn't going to be predictive enough from what we understand, so we won't be going forward to include that information into the genetic evaluations from what I understand at this point in time. So watch this space a bit still. But basically the two breeds you want to get the most out of it are, surprise, surprise, the two that have got the best reference populations with phenotypic information, which is Angus and Brahman. So again, it just goes to show that with that reference population and the closer you are related to it, the better outcomes you might get in regards to accuracy of molecular breeding values. The last point there to finish up on is, and this is getting quite technical now, but over time and over the next 12 months, I think we'll see a transitioning from what's called the blending approach to incorporate molecular breeding values. So at the moment, the way the breed plan analysis works, it just does its general regular genetic evaluation for what it does, and then right at the last minute, we'll find out which animals have got MBVs, and we'll just adjust the EBVs and the accuracy of those individual animals that have got the molecular breeding value on the database. What's going to happen in the future is this what's called the single-step approach, and it goes into storing the SNP profiles on the breed society database, and it's where we actually won't be incorporating molecular breeding values into the genetic evaluation from, well, I won't say we won't, but where the main source information is going to be coming from the DNA technology and the SNP profiles is the breed plan model itself will actually work out what values those SNPs are on an ongoing basis, and it's where it'll update regularly. The other thing that'll work on there is it's what's called this genomic relationship matrix. So pedigree is very important to the breed plan evaluation. We need to know who's related to who, but with the SNPs on file, we might get a better idea and a better picture of how animals relate to one another, not at the pedigree level, but actually the SNP level, which means we'll get a better idea of the relationship between animals. And from what I've heard thrown around is we should have a trial analysis based on the single-step approach available for one breed anyway over the next 12 months to have a look at to see how it works. So that's really where it's transitioning, and it really highlights to me the importance of getting the SNP profiles recorded on the breed society databases. So when this single-step approach comes in, we can get that all working correctly, and we've got a lot of information there already built up to work on. All right, so that's what I think the future holds for this whole space. So I'll be interested to come back in 12 months' time to see where that ends up. So I'm going to finish up the presentation there, but we'll definitely answer a few questions, and I know Gemma wants to finish off with a poll, so don't duck off so quickly. Alex, do you want to finish? Do you want to comment on anything I've said there in regards to what does the future hold, in your opinion?
Yeah, no, you're pretty well. I would agree with your analysis of what's likely to happen in the future.
Okay.
That's particularly Horn Pole, you got that exactly right.
Yeah, thanks, Alex. I'm glad that. Okay, Gemma.
Yeah,
are there any questions that we want to go through before we finish off? Maybe just one or two.
Yeah, just one question. I suppose it's probably a bit of a big one actually. There's a few breeds that have beef information nucleus herds going at the moment. Are they contributing at all to these DNA technologies, and if so, how?
I think I'll hand that question over to Alex, who's in a breed who actually runs a bin program, and he can comment from his perspective, which I think is pretty important. I've got some comments, but I think it's good if Alex can comment on that.
Yeah, thanks, Christian. Well, the first answer is that although we're only halfway through our bin, we've already used the data out of that to test the CRC predictions for limousin, and while the outcome wasn't positive, at least we got an answer as to whether the CRC crossbreed predictions were going to work for limousin. So already we've got important information, and as Christian suggested, as we move into this single-step analysis, the more animals that have both a SNP profile and also have very good phenotypic measurements, then the better the one-step analysis is going to work. So it's still early days, but the one thing that no one argues with is that the more animals you get with accurate measurements, particularly on the hard-to-measure traits or the carcass traits, which are also genotyped with either a 50K or even maybe a 10K, then the more likely then that SNP technology is going to work for that breed.
Yeah, it definitely puts those breeds in a better position going forward to have the technology available. That's for sure. There's no doubt about that, because it comes back to that reference population and having one that's related to the, or a validation population that's related to the population they're selecting from, which I think's pretty important to consider. Okay, Gemma?
Sorry, just that one of the reasons that Angus already has prediction equations that are quite useful is that they have the best database of measured animals with genotypes.
Yep, spot on. Gemma, have we got another question to finish off?
No, that's pretty much it at the moment. I think everyone's obviously understood where we're going with a lot of it, so that's good.
Great. Well, thanks, Gemma, and thanks everyone for tuning in tonight. I'm now going to hand over to Gemma, who will just finish off the presentation. I also want to thank Alex for his comment tonight. It's been very good, and I'm sure everyone's appreciated it. So I'll now just hand back over to Gemma, who can now finish off for this evening.
Okay, thanks very much, Christian, and thanks, Alex, as well.